are going to talk about a topic that I find extremely interesting, sensation and perception, which this might be one of those things that you probably have never really thought of as being in the realm of psychology, especially if you tend to picture psychology as like just therapy or counseling, then this might be a little bit surprising to you. I would also say, this topic is kind of what I studied the most in graduate school, so I could talk about this stuff for literally like hours. In my previous institution, I had a semester-long course called Sensation and Perception, so I spent 16 weeks talking only about this stuff. So sometimes it's a little hard to figure out like how much detail should I give you in an intro-level class. So if you want any more detail than what I give you today, feel free to ask questions, stop by my office hours, talk to me about it, because I find it really interesting. So before we get started, so sensation and perception, we're basically going to be talking about how it is that we perceive the world, how we see, how we hear, all those kinds of things. So how many senses do you think we have?
21.
Let's start there. What was that?
21.
21, that's a huge number. How many were you taught in elementary school that you have? 5, right? What are those 5 basic senses? Sight, hearing, taste, smell, touch, right? Those are the 5 that you're taught in elementary school. So I don't think there's 21, there might be, I don't know. It kind of depends on how we start parsing out senses, but there definitely are more than 5. So we can think about quite a few others. For example, your sense of balance. That doesn't really fit in with any of those 5 senses. There's a whole different sensory system that's devoted to balance that has nothing to do with any of the other 5. Even touch, like if we think about touch, it's actually separated into multiple senses that you might call all of them touch, but they're actually very separate. So probably when you think of touch, you're thinking about like right now if your neighbor reached out and poked you, you could feel that. Or if you're holding a pencil or something, you might be able to feel that pencil in your hand. That's probably what you think of with touch. But you also have like thermoreceptors that can sense temperature. You have different receptors that can sense pain that are totally different from those touch receptors. So we actually have a lot more than 5 senses. For this class, we are mostly going to focus today on vision and hearing, and then I'll talk just really briefly about some of those other senses. But again, if you want more detail about any of them, just let me know, and I can add more material, or it's something we could just talk about one-on-one in office hours if you're really interested in. So we have a lot more than 5. Let me ask you another question, and for this one I don't necessarily want you to answer out loud, at least not yet. I want you to just think for a minute if you have ever hallucinated. Okay? Now I'm going to guess most of you probably have not. Maybe you have if you've been really sleep deprived. Sometimes that can cause hallucinations. A little bit of an aside, but whenever I was in 8th grade or something, me and my stupid friends would sometimes stay up for 48 hours straight on purpose just to make ourselves kind of trip out and hallucinate. I never had a full-blown hallucination from doing that, but you might start to see, things kind of shift in weird ways, or your visual perception starts to get kind of weird. So if you've ever been really sleep deprived, you might have hallucinated then. If you've experimented with various substances, you could have had a hallucination under those conditions. If you're suffering from some sort of psychological disorder, you might have had a hallucination. But outside of those very extreme examples, probably none of you have ever hallucinated, right? So I'm going to kind of make the argument today, though, that really, pretty much all the time, you're constantly hallucinating if you really think about it. So first, let's maybe come up with what is a hallucination. Can anybody give me a definition of what you think a hallucination is?
A figment of the imagination.
Okay, a figment of your imagination. Let's make it a little more precise. I like that. So I think you're basically getting into the idea that a hallucination is where you're perceiving something that's not real, right? But right now I can imagine a pink elephant, so it's a figment of my imagination, but just because I'm imagining it doesn't necessarily mean I'm hallucinating, right? So maybe can we take it a step further than that? Anybody want to add to that definition? Make it a little more precise? So we know hallucination is where there's something that's not real. I would say the thing that you would add to that is that most people, when they're having a hallucination, it seems to be real, right? You're perceiving it, you're either seeing or hearing something, and your brain thinks it's real even when it's not, okay? So by that definition of a hallucination, I would say most of the time we're hallucinating, at least to some extent. And probably, that might sound crazy right now just for me to say that, but by the end of today's lecture, I hope you see that that's kind of true. So I'm going to give you one example right now of what we could potentially call a hallucination. So I'm going to show you this image. On the next slide, there's going to be a picture of a flag, but it's like weirdly colored. And I want you to just stare at that flag for 30 seconds. There's like a little dot or a cross or something in the middle. Just stare at that cross. It'll be there for 30 seconds, and then it'll disappear. Once it disappears, I want you to just keep staring at the screen, okay? Maybe. My PowerPoint is freezing. Give me a second here. There we go. So just stare at that dot in the middle for 30 seconds. You can blink if you need to, but try not to move your eyes around. Try to keep your eyes focused right in the center on that dot. Just keep staring. Like I said, blink if you need to, but do not move your eyes. It should disappear on its own, but it was frozen just a second ago, so it might. Let's give it like 5 more seconds, and if it doesn't disappear on its own, then in 5 seconds I want you to just look away and try to find like a white space, like a wall or a dry erase board. So you can go ahead and do that now. Just look away from the screen. Look maybe like over here where it's white, or over here. So whenever you did that, when you look away from this screen, what did you see? What happened? You still see the flag? Was there anything different about it? Yeah, so what you probably should have seen is right now it's up here in green, black, and yellow. Whenever you look away, you should see it in the opposite colors, which would be the correct colors of the American flag. So the green should turn to red. Do you? Interesting. The yellow should turn to blue, and the black should turn to white. I will say sometimes, so I have a lot of these kind of demonstrations of different illusions today, and sometimes they don't work super great on a projector. It just has to do with like the brightness of the screen, and it also kind of matters like where you're positioned and what your angle is and all kinds of things. So you might not experience exactly what I say, but just know that most people under the right conditions tend to experience what I'm talking about. So how many of you did see the flag kind of in the correct colors whenever you looked away? Okay, most of you did. All right, so I'll talk in a second about exactly what's going on, like why that happens, what's happening in your eyes and in your brain that makes that happen. But for now, I just wondered, like, would you consider that a hallucination? When you look away, you're no longer looking at the screen, the flag doesn't really exist over here, and yet you still see it. Do you think it's fair to call that a hallucination? You're shaking your head, why not?
Well, it's not really, for me, I don't think it's a hallucination. It's more like because of the brightness or like the colors makes your eyes just like kind of hang on to it.
Right? I would say it makes your eyes see something that isn't there, right? So by our earlier definition, like you're telling me the cause of why you're seeing something that isn't there, but you're still seeing something that's not real, and you're seeing it as if it's there, floating in the world. So, I don't know. Yeah, Cambry.
I would say that it's not, because he said that you think it's real and we know that it's not.
Okay, that's interesting. So yeah, so once you realize like, okay, this isn't real, then maybe that's what sort of breaks the definition of hallucination. I think I can accept that. Okay, let's keep going though. So for now, we can say that wasn't hallucination because you're all aware that it wasn't real. So, okay. All right, so now let's get into some actual content. Like I said, we'll come back to some of these illusions, and I'll show you some similar things in just a second. But in general, some of the main things I want you to take away from today is to understand that our perception, basically what's happening all the time when we're perceiving anything, when we're seeing something, hearing something, etc., we're taking some sort of energy from the environment and translating that into kind of a message that our brain can make sense of. So energy, in this case, I'm using that in a very vague, kind of broad way. So for example, light would be a type of energy. Sound waves would be a type of energy. Pressure on your skin would be a type of energy. And our sensory organs are basically just taking in all that energy and trying to convert it and make sense of it so that we can navigate the world. Like I said, we're going to focus mostly on hearing and vision, which obviously use light and sound as their energy. And light and sound are waves that have very specific properties that kind of contribute to the way that we perceive them. So we're going to talk a little bit about sort of the wave nature of light and sound, getting into physics a little bit, which is maybe weird in a psychology class, but you need to understand a little bit of that just to understand how our eyes and our ears translate that information and make sense of it. And then, like I said, we'll end by talking about some of our other senses. So we'll talk about some of the ones you named, like taste and smell, but we'll talk about some of the others that you maybe don't necessarily think of as senses. Just very briefly, we'll talk about those. All right. For some reason, my PowerPoint is wanting to update, so it keeps having issues. So just bear with me with that throughout today's lecture. Hopefully it doesn't cause any issues with the recording. Okay, so let's first talk a little bit about this idea of light and sound waves and kind of their properties. So first let's talk about light. So the reason you are able to see is because photons of light, which photons are just these little tiny packets of energy basically, are traveling and kind of, colliding with some cells at the back of your eye in a place called the retina. And so we need to understand a little bit about light to understand how that works. So first of all, know that the visible spectrum light that we can see is a very, very small sliver of what's called the electromagnetic spectrum. So the electromagnetic spectrum encompasses a bunch of other waves that we cannot perceive with our eyes. So for example, it includes things like X-rays and microwaves and gamma rays. And on this spectrum, the part that we can see is very, very tiny and narrow. So it's like this little tiny sliver that we have actual access to with our eyes. And essentially these waves have two main properties, are frequency and amplitude. So the frequency is basically how many times the wave passes in a given time period. Usually we talk about it in terms of seconds. So how many times does the wave repeat itself in 1 second would be the frequency of the light wave. So down here at this end where we're talking about, like gamma rays, they have very, very, they repeat many times in a second. So, we would call that a larger frequency. So you can see like the peaks, right? Maybe there's 3 peaks per second, whereas over here, as we get to the broader ones, there's only 1 peak per second or whatever. And so that frequency is going to determine the color of the light. So we have blue and sort of ultraviolet on one end of the spectrum, and then red at the other end of the spectrum of that kind of visible spectrum of light. And again, that light is just carrying energy. And typically, the higher the frequency, the more energy it's going to carry. Again, we'll go into a little bit more detail about how color perception works in just a second, like how your eye translates those different frequencies, but just for now, know that light is traveling at these separate frequencies. And sound works similarly. So sound travels in waves. Basically, as I speak, my vocal cords are vibrating the air around it, and then those vibrations are kind of sent out into the world through the air. Eventually, those vibrations make their way to your ear. And again, those vibrations are basically, we can picture them as being like waves that have different frequencies. And those frequencies, when we're talking about sound, they correspond to pitch, like how high or low the note is. Human hearing, again, has a very limited range, just like with vision. So we can hear between about 20 hertz and 20,000 hertz of sound waves. So again, 20 hertz would mean that wave is repeating itself 20 times per second. 20,000 hertz would be it's repeating itself 20,000 times per second. And this range is really kind of like what you're born with. And as you get older, you start to lose, especially like the higher frequencies. So most of you, if you're in your 20s, maybe still have that 20 to 20,000 hertz preserved. It kind of depends. Like if you've been to really loud concerts multiple times, if you listen to your headphones at their maximum volume really often, then you're going to lose those higher frequencies sooner. But if you don't do things like that, if you've kind of kept, good care of your ears, then you might still have that full 20 to 20,000 hertz. Other animals, you might know, have very different ranges, right? So like dogs, for example, can hear much higher pitches than humans can. So there's certain sounds that they can hear that we cannot. I have 2 dogs, and there are often cases where like maybe something's making a sound that I can't perceive, but I can tell that they can because they're reacting to it in some way. And then, there's other extremes. So like elephants, for example, can hear really low sounds. So they can hear stuff way below that 20 hertz. They can hear vibrations that travel like across the ground from even miles away because they're very low frequency and they kind of vibrate the whole ground. So again, kind of different species are attuned to different ranges for sound. So the frequency of the sound equals the pitch, like how high or low the note is. And then the amplitude, we didn't talk about amplitude, but the amplitude is basically how tall the wave is. So if we measure all the way from the peak to the trough of the wave, that would be the amplitude. And in general, amplitude corresponds to the volume of the sound. So the larger the wave, the louder it's going to be. The smaller the wave, the quieter it's going to be. So either way, with hearing and with vision, we have these types of energy that are traveling in waves, eventually making their ways either to our eyes or to our ears, where we will actually start to perceive them and start to make sense of them. So let's go a little bit more detail into vision. So vision, obviously the sense organ that we're going to be talking about is the eyes. The eyes are the organ that are going to take in the light and start making sense of it. And I tend to kind of think of the eyes as 2 parts. You have like the front of the eye and the back of the eye. And the front of the eye, the job is really just to take the light and focus it onto the back of the eye. And at the back of the eye, you have the retina, which is where there are actual neurons that are going to start processing that light and making sense of it. So at the front of the eye, all the structures, like I said, are dedicated to focusing the light. So at the very front of the eye, the first structure that the light is going to encounter is called the cornea. The cornea is this kind of dome-shaped, clear structure at the very front of the eye. And the cornea is going to start focusing the light. So it's shaped in that kind of dome shape because it's going to take the light and it's going to focus it onto the retina. If your cornea is misshaped, that can cause all kinds of vision issues, and that's essentially what like glasses and contact lenses correct for. And there's some other things that cause vision issues we'll talk about in a second, but that's one of the issues if you are like nearsighted or farsighted. It has potentially to do with the shape of your cornea. Another thing to know about the cornea is that it's rigid, so it cannot change its shape. So the light first goes through the cornea, then it's going to go through the pupil. The pupil is like the black hole in the middle of your eye, and that's just a hole that lets the light in. And around the pupil is, of course, your iris. That's the colored part of the eye. And the iris can open or close. It kind of controls the diameter of the pupil. So in general, whenever it's really bright out, our iris is going to close our pupil so that it's smaller. So we're taking in less light. It helps prevent the retina from being damaged from having too much light. And then if it gets darker, your pupil is going to get bigger. The iris is going to expand to make it bigger so that you're taking in more light so that you can see in like dim conditions. So the light goes through the cornea, through the pupil, and then eventually it's going to go through the lens. So the lens is right behind the pupil, pictured here. And it's this kind of like oval, spherical shape structure that again is clear. It's kind of similar to the cornea. The lens also helps focus the light, but the difference is that the lens can change its shape. So there's little tiny muscles that are connected to the top and the bottom of the lens that can kind of pull on it to either stretch it so it's like more oblong, or they can kind of release it and let it be more of a spherical shape. And that basically changes the way that the light focuses. So if I'm looking at something up really close to my face, and then shifting to looking at something far, it's the lens that's changing its shape to allow me to focus at those different distances. So the cornea stays the same shape. The cornea is really rigid, but the lens is flexible and it can adjust depending on what you're looking at. So the light goes through all these structures, and then eventually it's going to get focused on the retina. Okay? And the retina is where kind of the action of actually perceiving something starts. Any questions so far about kind of the front of the eye, structures there and what their purpose is? Okay. So, like I said, the front of the eye focuses the light so that it eventually makes its way to the retina. And the retina covers the entire back of the eye. It's kind of like a sheet that's over the whole surface of the back of the eyeball. If we were to like cut through that sheet and take a cross section, it would look like this image that I have here, okay? So this is like we're taking a cross section of the retina and looking at it and the different layers that make it up. And the way this picture is oriented, the light would be coming from the top. So the front of the eye would be at the top, the light's coming down and shining into the retina. And a couple things to notice about the retina. One is that it's very highly organized. It has different kinds of neurons that are arranged in these really precise, organized layers. There's basically 3 layers that have different kinds of neurons in them. The first layer, well, there's kind of 2 ways we can talk about this. I'm actually going to start at the bottom with these cells called the photoreceptor. So even though the light is coming through this way, it's going to kind of just ignore all these neurons up at the top, just kind of go through them, and eventually the actual photons of light are going to hit these cells at the very back of the retina. And those cells are called photoreceptors. And the photoreceptors are going to receive those photons of light, and there's basically this kind of complex chemical reaction that happens. Whenever a photon hits those photoreceptors, it causes them to basically change their shape and eventually fire like an electrical potential. So if you remember, I think it was last time we talked about like the structure of neurons, talked about dendrites and axons and all of that. The photoreceptors, they are neurons. They don't necessarily have dendrites really in the traditional sense. They have a very different kind of shape and structure to them. But they receive those photons of light and then they create this electrical potential in their axon. So then from the photoreceptors, and those are the ones here in blue and green towards the very back, those will connect to a group of cells called bipolar cells. So they're just sending information to the bipolar cells. Bipolar cells send information to the ganglion cells. And then the ganglion cells connect all the way to your brain. Okay? Just quick little summary of this slide. So the light travels through the layers of the retina, hits the photoreceptors. The photoreceptors basically convert that into electrical energy that then gets sent to the bipolar cells and then to the ganglion cells. I maybe should have an image here to kind of show you what I'm about to describe, but the ganglion cells, all their axons kind of bunch up into like one long cable or one long nerve called the optic nerve. And that nerve has to exit your eye somewhere. And where it exits, you actually have a blind spot. So in each of your eyes, there's a blind spot that does not see anything. Let me see if I can maybe kind of draw what I'm talking about. So if you imagine like we take the retina and we just lay it out flat. It's actually like a dome structure, but we're going to lay it out flat so it's like a circle at the back of your eye. Somewhere, it's not quite in the middle, but somewhere you have this little area, like there's a bunch of ganglion cells, right, like spread all over this. And they all have their little tails that kind of bundle up and then go to this area of your blind spot. And then from there, it's going to exit and make its way to the brain. So you have a blind spot in each of your eyes. Does anybody know why? Like right now, you can't see it. It's not like you look out in the world and there's a spot of vision just missing. Does anybody know why that's the case? Yeah.
Is it so you don't see your nose?
That's something different. I know what you're talking about, but that's not related to this. So when I say blind spot, I mean literally like there aren't, because the ganglion cells are exiting there, like this whole area doesn't have any photoreceptors, doesn't have any bipolar cells, doesn't have any ganglion cells, like it's not receiving any light. And yet we don't notice that blind spot.
Is it because the light's bouncing off?
No, good guess though. You keep making guesses. Yeah, Jesse.
Does our brain just fill in that spot?
That's part of it. So one thing is that our brain is just like, hey, I know that there's something there. There's no way that there's just nothing there. So I'm just gonna like guess and kind of fill it in. So your brain basically just takes like all the stuff that's around it. Like if I'm looking at this wall, everything is white, you know, ish or whatever. So my brain's gonna be like, okay, this blind spot is probably also white. The other thing that helps is that you have two eyes that are almost always open at the same time. And these blind spots don't like quite overlap. Like the blind spot of where your right eye is and where your left eye is are going to be slightly different. So then your brain can also use that. It's like, okay, well, I know that this part of my right eye isn't seeing anything here, but my left eye is seeing that, so I'm just going to take the information from the left eye and fill in this little patch over here. But then even if you have one eye closed, then that other mechanism where your brain just kind of guesses and figures it out is also filling it in. There are ways to test if you're interested, if you want to prove that you have a blind spot. There's like a very simple test. You can probably find it online. I could maybe even recreate it here, but it's like something where there's like a dot and an X, and you're supposed to like focus on the X and then move your paper until like the dot eventually will make it to where it's like just right in your blind spot, and the dot will like disappear from your vision for a brief second. So at some point, you can maybe look that up, look up blind spot test or something, and you'll probably find it. Let's see, what else do you need to know about the retina? So light hits photoreceptors, goes to the bipolar cells, the ganglion cells. Ganglion cells exit and eventually will make their way to your brain. We'll talk in a second about what happens in the brain, where it goes, and all of that in just a minute. But first, let's talk a little bit more about these photoreceptors, because you might notice on that picture there's like blue and green. There are two different types of photoreceptors. And those two different types are called rods and cones. And those names come basically from their structure, sort of how they appear. So if you look at like a rod, it's actually shaped kind of like this, the top of it, and then there's like the rest of the neurons goes out or whatever. Whereas the cone has like almost this triangular kind of shape to it. So the name just comes from their actual physiological shape. So there are these two different kinds. They both respond to light. So both of them, basically the photon of light hits it. Again, there's this complex chemical reaction that happens that causes it to fire electrical potentials. But they respond to different frequencies of light in really specific ways. So rods in general are basically like colorblind. They don't really care about the frequency of the light. They'll kind of respond to just about any light. But rods are more sensitive. So in general, it takes just one single photon of light to activate a rod, which a single photon of light is like a teeny tiny minuscule amount of energy. It's like basically nothing. So rods are very, very sensitive. So that means they're good at contrast, and they're also good at seeing in the dark. So if you ever, you wake up in the middle of the night, you need to go use the bathroom. Usually if you like first wake up, you don't turn the lights on, it's gonna be really dark and hard to see. But after just a few minutes of maybe navigating around in the dark, you eventually get to a point where like suddenly you can see more. You guys all know kind of that experience I'm talking about. That's because that's basically like your rods kind of waking up, essentially. Once your rods have woken up and they can receive the light, then they can see in really dim light conditions. So that's your rods. Your cones, there's actually three different kinds of cones. So we can break it down even further. So we have photoreceptors. You have two basic kinds, the rods and the cones, but then the cones we also have, three different kinds. And those three different kinds of cones all respond to different frequencies of light in different ways, and they are what allows you to have color vision. So there are some cones, for example, that'll respond really vigorously to like shorter wavelengths, and some that'll respond to a medium wavelength, and some that'll respond to a higher wavelength. So as those different frequencies of light are hitting your photoreceptors, your brain is kind of comparing the activity across those three different cone types to try to figure out what color something is. It's kind of almost like if you ever play around on, Photoshop or Illustrator or any like photo editing, you can input like an RGB value. It's sort of like that. It's like telling your brain like, hey, there's 25 red and 250 green and 0 blue or whatever. That's going to be a specific color based on those three combinations of values. So rods are really good for contrast and seeing in dark light. Cones, you have 3 different kinds, and they are what contributes to your color vision. Are any of you don't have to answer this question if you don't want to, because this is maybe like personal information, are any of you colorblind? Okay, so typically, there's colorblindness actually is different in males and females. About 12% of males are colorblind. For females, I think it's like, I don't know, maybe 5% or less or something. But usually someone who is colorblind, they are what's called red-green colorblind. And the reason they are red-green colorblind is because one of these 3 cones is basically mutated to the point that it's non-functional. So something in the gene of that person makes it so they're not producing that 3rd cone correctly. So they only have 2 cones that they can use to distinguish color. So then their brain basically doesn't see, they don't distinguish the same number of colors that we do. So if they were looking at like this color spectrum right here, the red and the green would basically, they wouldn't be able to see that. So instead, this green might be, kind of further colors of blue, and the red might be like further colors of yellow. So they can still see blue and yellow, but they can't see red and green, which is also the same physiology that dogs have in their vision. So you might have heard that dogs are colorblind, and most people tend to assume that they can only see in shades of gray, but actually dogs can also see blue and yellow. There are some species and even some people that actually have 4 cones instead of 3. So there's a very small number of people. For some reason, I haven't looked a ton into this, but for some reason, all the people that they found that have 4 cones are like women in some very specific area in Britain or something. There must be some sort of genetic pool there that allows them to have those 4 cones that hasn't spread to the rest of the population. But basically, if someone has 4 cones, then that means that they can see more colors than you and I could see. So someone who had 4 cones, when they look at this spectrum here, like to me, kind of all of this section-ish right here looks the same shade of blue. Someone that had 4 cones, they probably would see like 20 different color shades within that small little patch. So they can actually see more colors than most humans can. Does anybody know what the largest number of cones has been found in a different species? So there are other species, like I said, dogs have 2. There's lots of birds that have 4, just like the people I was talking about. Does anybody know any species that has more than 4? There is, so the largest that I know of, there might be others that have been discovered since then. There is something called a mantis shrimp, which is like a sea animal. It's not a praying mantis and it's not a shrimp, but it kind of looks like both of those combined, so that's how it got its name. They're these very brightly rainbow colored things that live on the ocean floor, and they have 12 different cones. So I can't even, if you try to imagine what their visual world is like, it would be completely different from ours because they can see just a ridiculously huge number of colors that we cannot perceive, basically. Part of the other thing, so sometimes when a species has another cone, they can just see more colors within this spectrum. But then some species also have cones that let them see ultraviolet or infrared light. So some species can see ultraviolet light that we can't perceive at all, or infrared light that we also cannot perceive. Okay. Any color so far about color vision or about the differences between rods and cones? All right, so again, from the photoreceptor, the information goes to the bipolar cells and then to the ganglion cells, all within the retina. Then the ganglion cells send their axon to the brain. The first place that the information gets sent is a structure called the thalamus. And the thalamus is kind of in the middle of the brain. And the thalamus, I always kind of think of it almost as like a little post office within the brain. The thalamus receives information from all your sensory organs. So it's going to receive visual information, hearing, all of that. And it kind of parcels it all out, makes sense of it, and figures out where it needs to go. So it's okay, this information is coming from the eye, that must be visual, I'm going to send it to the visual cortex. Okay, this is auditory information coming from the ears, I'm going to send that to the auditory cortex. So it's just kind of sorting things out, sending them where they need to go. So then from the thalamus, that information is then sent to what's called the primary visual cortex, which is at the very back of the brain, an area called the occipital lobe. So this image over here kind of shows how that happens. Another thing to notice from that image is that pretty much everything in the brain gets sort of crossed to the other side. So in general, your right brain perceives everything on the left side of your body and vice versa. And it's the same with vision. So the right side of your brain is processing the left visual field. So everything you see on the left side gets processed in the right side of your brain and vice versa. So the information goes from the retina to the thalamus, from the thalamus to the primary visual cortex. And the primary visual cortex is kind of starting to make sense of some really basic visual things. The primary visual cortex is going to start, for example, it's going to figure out where like the borders of things are. So as you're looking out into the room, your primary visual cortex is kind of parsing things up into like lines and borders, and maybe starting to make sense of shapes, like figuring out, okay, there's a rectangle there, and there's a circle here, and sort of putting all those shapes together. And then that information is going to be sent into two sort of parallel streams. So from the occipital cortex, that information is going to go up into the parietal lobe and also down into the temporal lobe. And those two different streams are doing two very different jobs simultaneously. So the one that's down, the one that's going down to the temporal lobe, is processing kind of the identity of the object. So it's figuring out that's a table, that's a chair, this is a marker, that's a thermos, whatever. It's figuring out what the object is. So that's the temporal stream. And then the one that goes up is figuring out spatial information. So it's figuring out, okay, that's in front of me, that's behind me, that thing is next to that thing. So it's figuring out kind of the location of different objects. One of the ways that we know about these two different streams is through experiments that were done on monkeys in, I want to say, like the 1960s, where they basically, it's kind of horrible, I know we talked a little bit about like animal research, and this is one example of it. They basically trained these monkeys to do two different tasks to show that they could recognize objects and they could recognize where the objects were. So like in one task, if they picked the cube, they would get a treat. So they had like a choice between like a cube and a cylinder or something like that. If they picked the cube, they get a treat. In another task, there was like two little holes in a table, and if they picked the hole that was next to some landmark, then they got a treat for that. So that's more like a spatial task. So they train these monkeys to where they can do both those tasks with like 100% accuracy. Then they damage one of those parts of the brain. So they either damage the parietal stream or they damage the temporal stream. And then they test the monkeys again on their ability to do those tasks. So if they damage the temporal stream, the one that's for object recognition, they cannot do that object recognition task anymore, but they can still do the spatial task. And then if they damage the parietal stream, they show the opposite. They can still do the object recognition task, but they can't do the spatial one. So that's part of how we know that we have these two different streams of brain that kind of process visual information in different ways. And then in humans, we've kind of confirmed that by like case studies. If you have someone who's had like a stroke or a car accident that affects one of those parts of the brain, they will also have these different types of kind of disorders where if there's damage to the temporal lobe, they might not be able to recognize objects. And if there's damage to the parietal lobe, they might not be able to tell where an object is located in space. All right, any questions about kind of how that visual information gets parsed out into the different areas of the brain? Okay. So I have now a few more illusions, kind of like the flag, that we're going to kind of demonstrate, and then I'll talk to you a little bit about what each illusion sort of tells us, basically. So first, I have a few that have to do with depth perception. So this first one, anybody want to describe what they see here?
Shadows.
Okay, shadows. Do those shadows give you any kind of like perception of depth? Yeah, I get that.
Bubble wrap.
Yeah, it kind of looks like those bubble wrap things, and it looks to me like some of them have been popped, right? So when I'm looking at it, it looks like maybe these ones have been like popped, so they're indented in, and then these ones are kind of like popping out towards you. Does everybody else see that? Okay. So I want you to kind of track. I'm going to make it so that this flips, so the ones that are at the bottom will then be at the top, and just kind of track with your eyes and see what happens. So when I flip it, it changes it, right? So the ones that look like they were popped in now look like they're popped out towards you. Everybody see that? Okay, so this is an illusion of your depth perception that is caused by shadows. So the shadows are very important here. Anyone in here like an artist, draw or paint, especially like realistically? Maybe know a little bit about shadows and highlights. Anybody know kind of what's going on, like why your brain perceives some of these, like obviously this is flat, right? There's no actual depth here, and yet your brain is perceiving depth. Anybody know why? Why does this one look popped out and that one look popped in? Yeah, Aiden.
Because it looks like the light is coming from the top.
Exactly, yeah. So if you look at this, the ones that have the highlight at the top look like they're popped out. The ones that have the highlight at the bottom look like they're popped in. And the reason is all because of the way that we perceive light coming from above. So if you think about it, for pretty much all of our evolutionary history, light would have always been coming from above us, because before we invented light bulbs and stuff, light only came from the sun, which was always overhead. And even now that we have electricity and light bulbs, we tend to put them in the ceiling above us, right? So almost always light is coming from above, and if light is shining down on something that's popping out, it's going to create this highlight at the top and a shadow at the bottom. And if it's on something that's popped in, there's going to be a shadow at the top, a highlight at the bottom, right? So our brains have learned to kind of perceive things that way just based on shadows and highlights. There was an experiment they did with chickens where they basically they had half of the chickens raised in like a normal environment where light was coming from above. The other half of the chickens, from the time they like hatched from their egg, they were in this environment where all the lights were on the floor coming from the bottom. And then they trained the chickens to basically peck at the ones that were popped out. And the ones that were raised in a normal environment, they would do just like we would. They would see that this is popped out and they would peck at it. But the ones that were raised where the light was coming from the bottom, they would peck at these and think that those ones were popped out. So they basically like learned to perceive depth differently based on where they were used to the light source coming from. So I didn't go into detail at all about how we perceive depth, but at least part of it has to do with cues like this, like with shadows and highlights, help us kind of perceive if something is, you know, above or below or popped in or popped out. This is another illusion of depth. So if you look at this image, these two, I don't know what kind of creature that is. It kind of looks like a beaver with its tail cut off or something. I don't know, but yeah. So which of these animals looks bigger? The one on the top or the one on the bottom? The one on the top looks bigger, right? In reality, they're actually the same size. Like if I cut that one out and put it here, they're exactly the same size. But we perceive the top one as being bigger. Anybody know why you think the top one is bigger?
Because of the lines of the railroad?
Yes, so the lines of the railroad create this illusion of depth. They make it look like the top of the image is further away than the bottom, right? Because of the way that the railroad is sort of receding. It almost looks like it's converging at like the horizon. So then it looks like that top one is further away. So basically to figure out the size of something, your brain is doing this calculation where it takes what I'm going to call the retinal size. So the retinal size is just like the actual size of that projected onto your retina. So it figures out, okay, what's the retinal size, but then it also tries to figure out how far away is the thing. Because if something looks a particular size, but it's really far away, then it's obviously bigger than what you're seeing, right? Like this marker is however big, but if I move it far away from my face, it's going to look smaller, right? So your brain's doing this constant calculation between like how big does it look, but also how far away is it? And if something looks far away, we're going to kind of assume that it's bigger than it actually appears. So this isn't necessarily illusion. This is just a picture to remind me to talk about this. It's called the moon illusion. So the moon illusion is where oftentimes it's called a supermoon. How many of you have ever experienced a supermoon? Where basically the moon just looks like ridiculously huge in the sky. And there's a couple things that cause this. Usually a supermoon is going to happen when the moon is really close to the horizon. So kind of, it has to be a full moon, and that full moon has to be visible at the horizon. Is typically what's going to give you kind of this illusion. And it also often helps if there's certain depth cues in the environment. So the most extreme I've ever heard that it is, I've never seen it myself, is like if you're out on the ocean and the full moon is at the horizon, then you'll get like the most extreme supermoon imaginable, basically. So the interesting thing, so like if we look at a moon at the horizon and a moon up in the sky, and if we were to, again, like kind of take pictures and measure the actual diameter of the moon, they're going to be exactly the same. But there's this illusion that's created whenever it's at the horizon that's kind of similar to the picture we just looked at. Basically, when something's at the horizon, you get all these cues that are telling you this thing is far away. Like if there's waves on the ocean, there's this pattern that happens where like things close to you tend to be really detailed, and things far away from you tend to be like more blurry. So if you have lots of waves on the ocean, you get this really pronounced sort of depth cue where like the waves close to you are really pronounced, detailed, the ones far away are going to be blurrier and smaller. And so it creates this kind of almost like the picture we looked at before where it's like making it look like the moon is further away. But when the moon's just up in the sky, there's none of those cues. It's just kind of in like open space. So there's nothing to tell your brain, hey, this thing is really far away. So you perceive it as smaller compared to when it's at the horizon. So very similar to the railroad thing, just happening kind of in a real world setting. So those are some illusions of depth. And I think the thing I would emphasize too that I maybe didn't say super clearly is that all these illusions tell us something about how our brain and our eyes are processing information. So we know a lot about the visual system based on illusions and how they kind of trick our eyes into seeing something that isn't really there. Okay, this one, a little more abstract. This one definitely depends a lot on like the projector and where you're sitting, so you might see something different. But does anybody want to describe? So the obvious thing here, we have different bars of different shapes. So there's a black one here, a white one there, and then grays in between. But does anybody want to tell me, if we look at just one bar, like if we focus on this one, what do you see in that one bar? Yeah.
So on the left side, where it meets the darker one, it looks lighter, and on the right side, it looks darker.
Yes, yeah. So for me, and hopefully for all of you, when I look at just one bar, it's not a solid color, it's like a gradient. So it's lighter here, and then it gets darker as you go that way. Everybody see that? Okay, since that's the thing I'm pointing out, you probably maybe are catching on that that's not real. So all I did here was just make some black rectangles in PowerPoint and put them over it. So when you cover up the neighbors, to me now this looks like just one solid gray color. Does everybody see that? So you only get the gradient when it has the neighboring ones. I could try to go back, but it's going to mess up my recording if I go back and forth. But yeah, so whenever you have the neighbors of different colors bordering it, you get this sort of gradient effect. But if you just cover those up, suddenly the gradient disappears. Now this one I find interesting. It's very like subtle, but if I start looking out into the environment, I can kind of almost find this everywhere. Like if you look at the walls and the corners where they like line up, and like one might be darker because it's in shadow than the other, I can kind of almost see like that like highlight at the edges of everything. And there's almost like this gradient everywhere. And I guarantee you, if I could somehow do this in the real world, if I could take like a black bar and just put it over things in the real world to do the same thing, where that gradient just like disappears if you just cover up the things that are neighboring it. Now this one, I'm going to tell you why this happens. This is a little more technical than some of the others, because there's like stuff actually going on in your photoreceptors that causes this illusion. And I think it's interesting. Might be a little in the weeds. So if like you get bored or this goes over your head a little bit, totally fine. But if you find it interesting, then I want to share it with you. So essentially what happens as you're looking out at that image or you're looking out at the world, there's all this light coming into your retina, hitting your photoreceptors. And there's different amounts of light, right? Like there's really bright lights that has lots of photons, darker light that has just a little bit of photons, and that energy is hitting your photoreceptors and making it fire kind of at different levels, basically. One thing you should know about your photoreceptors, this is true of a lot of neurons in your nervous system, is that they're kind of almost like competitive. So if one neuron is firing, it wants to like get all the resources, it wants to do all the firing, and so sometimes it'll inhibit its neighbors. So when a neuron is firing, it might kind of like tamp down its neighbors and be like, hey, I get to do all the firing, you don't get to do any, essentially. So if we zoom in, let's say we zoom into two of those bars that we were just looking at. So we have kind of a darker gray and a lighter gray here. And here I'm demonstrating like the photoreceptors that are receiving that information. So in your retina, there are some photoreceptors that are receiving light from the light bar, and then there are other photoreceptors that are receiving information from the darker bar. Okay? So these numbers are completely arbitrary, but it's just to demonstrate the point, basically. So let's say because this is brighter over here, there's more photons of light, so these photoreceptors are getting lots of energy. Let's say they're getting 100 units or whatever of energy, whereas the dark ones are only getting 50 units of energy because it's darker. So that's kind of their initial input from the energy of the light. But then they're going to inhibit their neighbors. When they inhibit their neighbors, though, they do it at kind of like a proportion of the energy they're receiving. So for this example, I'm going to say that they're inhibiting their neighbors by 10% of their initial input. So if this is getting 50 as its initial input, 10% of 50 would be 5. So all these dark gray ones are going to inhibit their neighbors by 5. All the brighter ones are going to inhibit their neighbors by 10 because they're getting 100 energy, and 10% of 100 is 10. So now we just do some very simple math. So for all of these, so like this neuron is getting 50, but then it's getting -5 from this neighbor, and then -5 from that neighbor, and we're just going to go across and kind of map all that out. So all of those are getting -5 from their neighbors. All of the ones that are getting 100, they're getting -10 from their neighbors. So then we get to this border, which is where the interesting stuff happens. So at this border, because you have a bright one and neighboring a darker one, the bright one's going to inhibit this one by 10, but the dark one's only going to inhibit its neighbor by 5. So then if we look at the actual output, so this output is the energy then that they would send to like the bipolar cells and the ganglion cells and eventually to the brain. So we just take 50 minus 5 minus 5 is 40. 100 minus 10 is 80. So that's kind of the extremes. But then it's this border is where we get the difference. So it's 50 minus 5 from this neighbor minus 5 here is 40. Oh wait, here, sorry, that's the wrong place. 50 minus 10 minus 5 is 35. 100 minus 10 minus 5 is 85. So now you got this discrepancy, right? Where this looks darker and this looks brighter. Does that make sense for all of you who managed to stay awake and pay attention to all that math? So, okay, the reason I get this much into the weeds here and go into this much detail is because I think the fascinating thing that's happening is, like, it's been demonstrated through all kinds of experiments that this is literally what's causing this illusion I just showed you, is this lateral inhibition in the photoreceptors. Keep in mind, photoreceptors are the very first layer of neurons that are receiving this light. And this math that they're doing, all your neurons are constantly doing very basic math like this. They're inhibiting and exciting their neighbors to different extents, which just adds up to different numbers, basically. But from the time the light hits your retina to where you're consciously aware of it, like you're not consciously aware of what you're seeing until it gets to your cortex. So it has to go through all those layers of the retina. It has to go to the thalamus and be processed there. And then it gets sent to the primary visual cortex where you start to become consciously aware of it. And then there's all that processing that's going on in the spatial stream and the object recognition stream. And every step of that is doing these little tiny calculations and kind of altering the reality of what you see. And if we added up like the layers of those neurons, it's probably at least thousands of layers of neurons that that information is getting sent through before you become aware of it. And the first layer is already causing you to see something that isn't really there. The first layer of calculations is causing you to see a gradient that doesn't exist. So if you start thinking about, okay, those calculations are happening times 1,000, then by the time you become consciously aware of something, you're not really seeing reality. You're seeing what your brain has decided is useful. Okay? So our brain's not just doing this arbitrarily for no reason. Can anybody think of a good reason why it might be important to kind of have this gradient effect?
Depth, the walking maybe?
Walking, definitely. Depth is part of it. So it's kind of contributing, I would say it's contributing to like spatial awareness. Aiden, do you have something?
It enhances the outline of objects.
Exactly. So essentially what this does is it makes it so that all the edges of things are highlighted. It makes it stand out. So when I look around the room and I see the table and I see the chairs, I can see these very clear separations between objects so that it's easy for me to interact with the world. I can walk around and not run into the table. I can sit in the chair and not fall on the floor. So because our brain is doing all this processing, it makes it easier to interact with our environment. So we're not really seeing what's real per se. Like if you could just measure the raw energy of the light, it's very different from what our brain actually ends up seeing. But all those layers of kind of unreality that's added is making things more useful. It's making it easier to interact with the world. So that's why I shared this one and get into the weeds, because that kind of fascinates me. How are we on time? Let's see. We'll do a couple more of these, and then we'll move on to talking about some other types of perception. Okay, here's one more. So as you look at this, obviously we have a bunch of black squares with white lines in between them. But what else do you see as you look at this image?
Each corner has a gray square.
Yeah, so you might see kind of like a gray, smudgy shape in the intersections. Anything else you notice about those gray, smudgy shapes?
They disappear when you look directly at them.
Yeah, you can't focus on them. They're kind of always like in your periphery, and if you try to look directly at one, it disappears. Everybody see that? Okay. This one's called the Hermann grid illusion. So basically what's happening here, one of the things I didn't tell you about your photoreceptors, another difference between rods and cones, is that cones have a smaller sort of like field of view. So they are more detailed, they have a higher acuity. So your cones are basically like mapping very tiny pinpoints of light in the environment, whereas your rods have a larger field of view. So they're taking in information from a larger pool of light, basically. Okay, so again, if we kind of like tried to map out the retina. So again, let's say we lay the retina flat, so it's a circle. Your cones are mostly concentrated in a part of your retina called the fovea, which is kind of roughly in the middle. It's not quite in the middle, but we'll put it there for the sake. So let's say this is your fovea. The fovea is where basically you just have a bunch of cones all bunched up together. And the cones are really, really packed super tight, and they're like tiny, and they're getting really tiny detailed amounts of information. I'm not going to fill that whole thing in, but you get the point. So you can kind of think your fovea is like really high resolution. If you think of like the resolution of a computer screen or something like that, okay? Then your rods are mostly located in the periphery. And the rods have like a bigger area that they're devoted to. So they're kind of more like this. And it goes all the way out to the periphery of the retina. So then whenever you're looking at this image, whenever you focus on something, you're basically making it so that light shines into your fovea, and your cones can see all these little tiny details. They can see all the little details of that intersection, but everything in your periphery, it's almost like it's blurring information together. So these rods, since they have that bigger field of view, they're kind of blurring information across like multiple areas. So your rods are basically, they kind of see that like there's some black and there's some white, but it just sort of blurs everything together and doesn't have a very high resolution. And then of course, when you focus on one of those gray blurs, you're shifting it so that it's now your high acuity fovea with all its little cones are seeing the intersection more clearly. Does that make sense? Okay. There's an image that kind of represents that. I'm going to skip past it. Okay, color. I think we have just 2 more illusions, and then we'll move on. We already did the flag thing, so when you looked at the flag and then it disappeared and they were opposite colors. Everybody remember that? I guess I can kind of describe why that happens now. So another thing that allows you to see color, there's like the cones doing their thing where they get the 3 different inputs or whatever. But then you also have your ganglion cells receive that information, and they kind of respond to things in this more like opposing way. So you have some ganglion cells that will be basically activated by red and inhibited by green. And you have some ganglion cells that will be activated by blue and inhibited by yellow. And some will be activated by black and inhibited by white. So they have this more kind of like opposite effect to the way that they sort of respond to things. The interesting thing though is that your neurons, they're just like any other cell in your body. They can get tired, they can get worn out. So whenever you were looking at that flag that was green, black, and yellow, and you're staring at it for 30 seconds, there's some ganglion cells in your retina that are seeing that black, and it's making them fire like crazy. Like they're just firing and firing and firing because it's black and black and black for 30 seconds. And then when the flag finally goes away and it's not there anymore, because those neurons have been firing so vigorously, they're actually going to drop their activity lower than normal. So there's kind of this baseline activity they're always at. And once they drop lower, that basically tells your brain that you're seeing white instead of black. And then the same with the green and the yellow. Your brain's firing super fast to the green. As soon as it goes away, those neurons kind of tired, they're fatigued, they drop their activity, your brain sees red, etc. So that's one of the color illusions. This other one, this next one shows kind of a higher level of color processing that we'll talk about in just a second. So do any of you remember this image, the dress, from, I don't even know what year this happened. But if you're not familiar, so how many of you, when you look at this, see a dress that is gold and white? I do. How many of you see a dress that's blue and black? Interesting, okay. All right, so this was it like took over the internet for, I don't know, a while, a few years ago. This has to be quite a while ago at this point. I need to find an updated one. But I didn't believe it at first. I remember seeing the image and being like, that's definitely a gold and white dress. Anyone who thinks it's blue and black is out of their mind crazy. They're probably lying for some weird reason. I don't know. I had all these theories about why would anyone say that. And then I called my son into the room, who at the time was probably 6 or 7 or something. And I showed him this and what color is this dress? And he said blue and black. And he had no internet access, nothing at that point, so I know he hasn't seen it yet. He really sees blue and black. I'm like, what are you talking about? You're crazy. So I see gold and white, and it's weird the distribution because as it was happening, it was about 50-50. Half the world saw gold and white, half the world saw blue and black, but most of you seem to see blue and black, which is interesting. Okay, yeah, Agatha.
I just see the gold on top. I don't know how you see white. Where's the white?
Okay, to me, this lighter color looks white, and this looks gold. So, okay, here's the interesting thing. If I were to take this image, again, on Photoshop or something, and do a little eyedropper thing, and go to a single pixel, and get the color, the real colors of the photo are, this is really kind of a goldy, brown color, and this is a very light blue. So it is blue, but it's very light. Okay, so that's the actual pixel by pixel color of the photo. Now, if you look at a much better picture of the dress, it is actually blue and black. It is a very dark, royal blue, and then all of these kind of frilly stripes are black. Okay, so everybody's kind of right, depending on how you look at it. I don't care about who's right or wrong for this demonstration, though. I want to know why. Why is it that people see this so different? Because it's kind of crazy. And the reason has to do with the way that our brain is interpreting the source of light. So when we look out in the world, we're not just getting photons and figuring out the color from that. Our brain, again, is doing all these calculations to try to figure out the true color of something based on the lighting and all this other information. So one thing you might not know is that this photo was taken with flash. So the flash was on when this photo was taken, so it was flooded with light from the front. Okay? But if you don't know that, you might think that the light is coming from behind because there's this huge bright patch right here. Okay, so to me, it looks like the light's coming from the back, but in reality, the light's coming from the front. And when your brain first looks at this, it very quickly makes a decision about which of those is true. Is the light coming from the front or the back? And based on that decision, it's going to entirely change the way your brain interprets it. If the light is coming from the back, then that means that this whole dress is in shadow, and it's darker than it is in reality. And so then my brain is going to be like, okay, well then I need to subtract all the dark values to get rid of that shadow to see the real color. If you take away the dark values, then you're left with white and gold. But if you interpret the light as coming from the front, which is the correct interpretation, then your brain is going to be like, okay, this is flooded with too much light, so I need to subtract all the bright values. I need to make it darker. If you make it darker, then you're going to see blue and black. So just that one tiny decision about is the light coming from the front or the back completely changes the way that you perceive the image. Does that make sense? Okay. All right, last one. And this one's kind of similar to the flag one. So this one's a video, but kind of the same. There's going to be a dot in the middle that I want you to stare at for 30 seconds. And as you're staring at it, there's going to be these kind of like bars moving downwards. So there's that blue dot in the middle. It hasn't started yet. These instructions go on for way too long. There we go. Okay, so stare at that blue dot in the middle. Same as with the flag. You can blink if you want to, but try not to move your eyes around. Try to stay focused on that dot in the middle. Keep staring at it, and eventually it will be replaced with something else. All right. So what do you all see as you're staring at this waterfall? It seems to be moving, right? And it probably looks like the water is moving down, like a waterfall would.
It didn't work for me.
What's that?
It didn't.
Yeah. So again, just kind of depends. If it didn't work for you, you might have unconsciously been moving your eyes without realizing it. Oftentimes we do that, especially if you're forced to stare at something, your eyes get tired. Let me see. I might be able to play it again without messing up my... I'll let it play again so you can try again while I'm talking if you want to. But essentially what's happening here, again, kind of similar to the flag. So in an area in your parietal cortex, you have cells that respond to motion. And they respond to it kind of like the cells I was talking about earlier. So there's some cells that if they see motion going upwards, they'll get really excited and respond a bunch. If they see motion going downwards, they'll be like inhibited and they'll respond less than usual. And so as you're looking at this, these bars are moving up over and over again for 30 seconds. And they're basically making those cells fire a bunch. And after the 30 seconds, those cells get tired. And so as the movement goes away, those cells are firing less than they normally would. And so your brain interprets that as, oh, these cells are firing less, this stuff must be moving down. So then you see the water moving down even though it actually isn't. So sometimes I have this happen in real life. You might have experienced this if you're driving a really long distance, especially at high speeds, and then you come to an intersection and stop. And so as you're driving, the world's kind of flying past you in a very specific direction. When you come to stop, there might be a moment where it sort of looks like the world is like drifting away from you a little bit. So if you ever have that experience, it's the exact same thing happening, just in the real world. Okay. That's all I have to say about vision. Any questions about that before we move on to our other senses? Okay. All right, so next we're going to talk about hearing. I have kind of less to say about hearing, mostly because once you understand vision and how it works, a lot of the other senses have kind of similar properties and similar mechanisms. So we'll talk about hearing, but you'll see there's lots of kind of metaphorical similarities to vision when we talk about hearing. So one of the first similarities is that the ear, very much like the eye, is kind of built up to take energy and focus it to a very specific organ so that that organ can then start processing the information and making sense of it. So your whole outer ear, what you probably think of when you think of an ear, is called the pinna. And the pinna is designed the way it is so that it will capture sound waves and kind of funnel those into your auditory canal. So the sound waves come in, pinna funnels them into the auditory canal. Once they reach the end of that auditory canal, they're going to hit your eardrum. The fancy name for the eardrum is the tympanic membrane. Those both mean the same thing. And your eardrum is just this very thin membrane that's going to vibrate as soon as those sound waves hit it. So it is very much like a drum. So the sound waves hit it, the eardrum starts vibrating. And then those vibrations are kind of amplified through these three little teeny tiny bones in the middle of your ear, called the malleus, incus, and stapes. Those are Latin words that basically describe the shape of them. So there's one that looks kind of like an anvil, one that looks like a hammer, and one that looks kind of like almost like a tuning fork. So the vibrations go through the ear canal, vibrate the eardrum, then they're going to vibrate those three bones. And as they vibrate it, the sound waves kind of get amplified and pushed into this structure called the cochlea. And the cochlea is kind of like the retina. So it's where all the neurons live that actually start processing that sound information. So the cochlea is this really complex structure. Here we're just talking about this part that looks kind of like a snail shell. It's like this spiraled thing. If we were to, let's say we take that cochlea, the snail shell, and kind of like unwind it. So it's almost not like a hose, basically, or like a long tube. And then we cut through that tube, and we're looking at a cross-section of that. Okay? Everybody follow me? So if we do that, if we cut through it, we're looking down the tube. It would look kind of like this. There's like this membrane here. There's a couple membranes. The important thing, though, is there's a bunch of fluid flowing through that tube. Okay? And so when the sound waves hit the eardrum, vibrates, gets amplified, pushed into the cochlea, all this fluid is going to also vibrate just like the sound waves do. And on this membrane, there are all these little cells called hair cells. And they're called hair cells because they have these really long, like cilia, hair-like structures on them. And they're kind of embedded in this membrane here. And they all have like all these little hairs. Okay, sort of like that, all of them, across them. So this fluid is vibrating, just like the sound waves. It's kind of sloshing back and forth. And as it sloshes back and forth, it's going to kind of displace those hair cells. It's going to cause them to move around in the fluid. So I kind of have that here. So again, these hair cells would be inside the cochlea, kind of on this membrane that's sort of hovering in the middle. So as the fluid comes in and it pushes and pulls on these hair cells, these hairs are connected to each other with these little structures called tipplings. I don't know who came up with that name, but that's just what they're called. And the tipplings are kind of like chains that are holding the hairs together. And they're actually attached to like receptors on the hair cells. So remember we talked a little bit about receptors. They're like the lock and key mechanism with neurotransmitters, all of that. So the tipplings are kind of connected to these receptors. So as the water is moving and it sloshes these cells and those hairs are moving back and forth, those little chains are going to push and pull on those receptors. So if it moves in one way, the chain's going to pull so that those receptors actually come open. And if it moves the other way, it's going to kind of relax and let that receptor close. And as the receptor opens, there's, again, a whole chemical process that basically happens that makes that hair cell fire an electrical potential. So again, just to kind of summarize, sound waves go into the outer ear, they get funneled into your ear canal, they hit the eardrum, which vibrates, and then those bones kind of amplify the sound into the cochlea. And then inside the cochlea, this fluid is moving like a wave that causes these hair cells to move back and forth, which causes the receptors to either open or close and causes them to fire. So then kind of like your retina, eventually, there's cells that send their axons to the brain. They will also send that information to the thalamus. And again, the thalamus, little post office, is going to sort it all out and decide that should go to the auditory cortex because it's auditory information. And then there's actually a very similar process that your auditory cortex uses, kind of like your primary cortex. The auditory cortex also then has two streams. One stream figures out what the sound is, like, okay, that's police sirens, or that's a cat meowing, or whatever. And then there's another stream that figures out where the sound is coming from. Is it coming from far away? Is it coming from my left or my right, or above or below me? So lots of similarities between vision and hearing. Any questions about hearing? Again, a lot more detail I could go into. For time's sake, we'll move on unless you have questions. Okay, the other senses we'll talk about in even less detail. I'm just going to kind of mention them a little bit and tell you just very briefly what's happening with each of these. So next we have what's called the chemical senses. These are often kind of classed together. So those would include things like taste, smell, and then what's called the common chemical sense. So you all probably know what taste and smell is. The common chemical sense is slightly different. Common chemical sense is basically what lets us detect like volatile, irritating substances. And there are cells or receptors for that in our mouth and in our nose, but also in our cornea, in our eye. And that is basically what allows you, it's what makes you cry when you're cutting onions, for example. So if you eat onions or smell onions, or if onion particles get into your eye, they all might have kind of like an irritating effect, basically, because of this common chemical sense. They're called the chemical senses. So if you think about the senses we've talked about so far, like vision is taking light energy and converting that into an electrical potential. Sound is taking sound energy, sound waves, and converting that to an electrical potential. The chemical senses are taking actual chemicals in the environment, and then there's interactions that convert those to an electrical potential. So with taste, smell, and the common chemical sense, you have to actually come in contact with the chemical in order to sense it. Anything in particular you guys want to know about taste or smell? Again, we could go into lots of detail, but I figured I'd focus mostly on vision and hearing for this class. Maybe think about it, and if by the end of the class you have any questions about taste or smell, let me know and I'll answer whatever I can. Okay, in addition to chemical senses, we have somatosensations. So somatosensations is probably what you think of as touch, but it's kind of a bigger umbrella that encompasses lots of things. So like I was saying earlier, you have touch, like pressure, just sensing if something's poking you or on your skin or whatever. You also have pain, itch, and then temperature receptors. All of those use very different mechanisms and different receptors. So kind of like photoreceptors and hair cells, use different types of energy. All of those also use different types of energy from the environment to make sense of things. Another somatosensation is called proprioception. Proprioception is basically our body's sense of awareness of like where our body parts are in the environment. So like right now you could close your eyes and touch the tip of your nose very easily. You're not using vision to do that. You're not technically using touch. You're using proprioception, which there's a lot of receptors like in your muscles and things that help you figure out where your limbs are relative to each other. And then the last one, vestibular sensation, that's your sense of balance. If I go back, again, I can't do it because it'll mess up the recording, but if you go back to the image I was showing you of the cochlea, you might have noticed there was like some other structures on there. So you have the cochlea is kind of like the spiral snail shape, and then next to it there's like these weird loop things that you might have seen. Your sense of balance is all happening in those loops called the semicircular canal, and that works very similar to your hearing. So all these loops are lined with all these little hair cells, and whenever you like move in the environment, if I tilt this way or that way or forward or backward, or even if I'm moving, the hair cells in these loops get displaced by that movement. So if I tilt one way, it causes the fluid in one of them to kind of spin around or move, and that also, there's also hair cells here, just like you're hearing, that get displaced and tell you if you're tilting from one side to the other. If you spin around in circles really fast, then one of these, the fluid will slosh around very quickly. So our sense of balance is actually very similar to our sense of hearing as far as the mechanism of how it works. Okay, those are all the ones I could think of. Anybody have any other senses that aren't listed here? Isaac said there were, what, 21, 23, something like that? So I don't know if there's others you were thinking of in addition to these. Any other senses? I think this is pretty much all of them. Okay.